Good morning, children. Hope that you all are keeping fine. This is the continuation video of chapter 4 of biology for class 10th students. Today we will be continuing with the topic uses of turgidity to plants. There are many uses. First one is turgidity provides rigidity to soft tissues such as leaves. So in leaves, when the leaves are in turgid condition or when the cells of the leaves are in turgid condition, it gives rigidity. If it is not turgid, then the leaves wilt. So this one you might have seen during a hot sunny day. If you observe a herbaceous plant leaves, you might have seen that is drooped down. It is because water is lost due to transpiration. During morning hours, especially afternoon hours, transpiration rate is more than that of absorption of water. So more water is lost, which results in the drooping down or wilting of the leaves. And in evening, again, restoring of the turgidity is attained by the cells of the leaves and it regain the rigidity. It is because the rate of transpiration is reduced than that of the absorption, or absorption of water is more than that of transpiration, so the cells regain the turgidity and it become again rigid and turgid, as so that the leaves again stand out during evening time. Another one important use is the turgor pressure helps to push through the hard ground. That one we can see in a germinating seedling as well as mushroom and all. From the harder ground or underground, it comes above the topsoil because of this turgor pressure. This is another one use which one we can see in plants. So here again you might have seen cracking of walls. When if a plant grow nearby a building, you might have seen the wall is cracked. It is due to the pressure exerted in the roots of this plant. That is the reason why. It is also due to the turgor pressure. The next one, turgidity helps in opening and closing your stomata. The structure of stomata you have already learned in class 9. Here, during the time of day or daytime, when photosynthesis takes place, the guard cells has chloroplast and there photosynthesis takes place. So glucose is produced and hence solute molecules, the concentration of solute molecules increase. So what happens? From the neighboring cells where the water content is more, that one is diffused into the guard cells and the guard cells become turgid, and which makes the guard cell to open. And the reverse happens in the at night time. Night time there is no photosynthesis, so the guard cell become, the solute concentration become very less as compared to the neighboring cell. So from the guard cell water is lost or water is diffused out, which results in the closing of stomata. So turgidity helps in closing and opening of stomata. That is another one use in plants. In plants, some movements are caused due to turgidity, or it is known as turgor movement. An example, drooping down of leaves of the sensitive plant, or touch me not plant, Mimosa pudica. Have you ever touched this plant? Of course, you might have touched sometime. So there, when you touch the Mimosa pudica plant, the leaves droop down. You might have noticed. It is because of the loss of turgidity in this petiole region. So here you can see a part of the plant where it shows the petiole stem and all. It is the condition when it is not touched, and here it is after being touched. So here the petiole, that is the stalk of the leaf, and a pulvinous region is marked. Pulvinous means the swollen region of the petiole. So when you touch the leaf, the pulvinous region of the leaf petiole become, or it loses the turgor, or turgidity is lost from the cells of this part, and hence it droop down. Some other interesting examples we can see in plant kingdom, such as the bending movement of certain flowers towards the sun, like the sunflower, and the sleep movement of leaves of certain plants during night. So these all are examples of Turgid movement in plants. Very important, usually used to ask in exams. Okay. Another one important point is that Turgid in root cells builds up root pressure. So this one we can understand with the help of this simple experiment. For this we need a well watered potted plant, but we have to cut the plant just some centimeters above the base of the stem. Then a glass tubing and a mercury manometer connected to it. So this is the experimental setup. So when you cut the base of the stem, water oozes out, or the cells are ooze out. Here it is connected with the glass tubing, so it enters into the glass tubing, which push the mercury towards up, or there is a rise in level of the mercury. Why does it happen? It is because of the root pressure created in the cells of the root. So let us understand the structure of root with the help of a diagram. Before that, this oozing out of cell sap from the injured or cut part of a stem is known as bleeding. Okay, that definition is also important. This diagram shows the cross section of a part of a root, showing the process of osmosis. So here first of all understand the structure. So here you can see the cells and many different types of cells. You can see the first layer of cell, this is known as epidermis. So the extension of epidermis is the root hair, already we have learned in the previous videos. And here many layers of cells you can see, these all cells comes under the cortex region of the cell, the root, and the innermost cortical layer is known as endodermis. Then there is another one layer that is known as pericycle. Then the conducting tissues, namely xylem and phloem. This is the structure, internal structure of the root. So when the root hair absorb water from the soil, water enters into the root hair and the root hair cell become turgid than the neighboring cortical cell. So water from here to here moves, or osmosis takes place, and this cell become turgid than the neighboring cell. So water from here moves to the next cell. Like that, water endosmosis takes or osmosis takes place from cell to cell, or cell to cell movement of water creates a pressure in the cortex cell or the cells of the root, and that is known as root pressure. And at last it enters into the xylem. From there it goes to the other region of the plant. Okay. So what is root pressure? It is the pressure developed in the root cells due to the inward diffusion of water through the cell to cell movement, or to the cell to cell osmosis takes place, which creates a pressure in the cells of the root, that is known as root pressure. And this root pressure helps movement of cell sap to certain region, or the upward movement. That is what we have seen in that previous experiment by using the mercury manometer. Then another one term related with the root pressure is of guttation. So guttation is the oozing out of water molecules from the margins of the leaves due to root pressure, excessive root pressure. So you might have seen certain plants like tomatoes, grass, banana, etc. in early morning. You might have seen there will be the droplets of water. This droplets of water comes due to the process of guttation. So in early morning there is less transpiration and root pressure is more. So water comes out of the leaf margins through the veins, and it is because of the process. That process is known as guttation. Okay. Now you have to recall the process of plasmolysis. In plasmolysis, what happens? Opposite of turgidity. That is, the water content of the cell is lost due to the exosmosis. When exosmosis takes place, when the surrounding solution is of hypertonic. So biological application of plasmolysis. Here in salting of meat or any other pickles and all, here the plasmolysis, the biological application or the biology behind that or the science behind that is of plasmolysis. So salting and making jellies, jams, etc. Their high concentration of salt or sugar will be there in the as the medium. So any bacteria, fungus and all, when it enters into this medium, their plasmolysis takes place and the bacterial cell or the fungal cell die, so that we can preserve the food material by this method. And also another one thing, weeds can be killed. Weeds mean unwanted plants can be killed in a playground by sprinkling excessive salt. Excess salt, when you sprinkle, what happens? The surrounding soil water become hypertonic, and from the cells of the plant water comes out due to exosmosis, which results in the death of the cell and eventually the cell death of the plant. So these all are the biological application of plasmolysis. Now we have some experiments based on absorption and conduction. That one we will be learning in the next video. Okay, thank you.